Sorry about that. But thanks so much, Shade, for joining us today. And over to you. Thanks.
Thank you so much, Rolanda, and happy to be here to present part of a pipeline we generated with a team from UCSF and the Makerere University, IDRC, and a couple of other institutions. So to start with, I'm going to present this tool, but in general terms, this whole idea is about Plasmodium multiplex amplicons for drug diagnostics, diversity, and differentiation haplotypes. So the tool of interest we are going to discuss today or focus on today is called MadHatter. I know it might have quite a sounding name, but it's a short form for multiplex amplicons for drug diagnostic diversity differentiation haplotypes via targeted resequencing. And this is mainly applied in a malaria parasite like Plasmodium falciparum, where the parasite is quite complicated compared to most of the pathogens commonly dealt with. So the focus of this discussion is mainly going to be on the design, which I will touch shortly, and then bioinformatic pipeline and the experimental performance. We have tried the pipeline and so far how it's being implemented or where it's being implemented. So to start with the design, this multiplex amplicon sequencing for Plasmodium falciparum microtypes, it's highly sensitive because it captures rare variants in polyclonal infections and in low density infections. So malaria parasites can be monoclonal, where it has one specific genotype infecting a single host, or polyclonal, where it can have multiple genotypes infecting a single person or a single host. So the pipeline is also using high throughput, whereby it can take in hundreds of samples in parallel, and it can be utilized in a multi-purpose way to look at the drug resistance in Plasmodium falciparum and other Plasmodium species, and the diagnostic resistance markers, diversity, and other parameters. And it's also created in a modular kind of format whereby the diversity is a panel, and there's a diversity panel, a diversity group of amplicons, resistance amplicons, diagnostic resistance amplicons, among others. So to dive in on the MadHatter pipeline, like I said, this is a multi-purpose tool. So the drug resistance markers focused on include, but not limited to K13, which is commonly known to be for artemisinin, and then CRT for chloroquine, among others. And also there is diagnostic resistance. So we capture the HRP23 deletions, which are commonly known to be diagnostic resistance markers in the RDTs. And also we have the CSP marker, which is a vaccine target for malaria vaccination. And then we also target 4 non-falciparum species identification markers or genes, the PV, PM, PV for Vivax, PM for malaria, PO for ovary, and the PK. And then finally, we have the genetic diversity markers, which are quite many, as we shall see. And these mainly are being used for multiplicity of infection, which is basically the number of specific genotypes in the single host. Infection relatedness, basically like IBD, identity by descent, and other things. So basically, this is to see how first of all, within a host, how different are the individual genotypes within a single host, and then also how different or related are the different parasites between hosts hosting the infections or between samples. Then it also used to do transmission networks, basically trying to show which parasite is connected to which parasite and which infection is connected to which infection. So this, in a simple terms, I think in the common pathogens, it's what we would have done like phylogenetic tree, but in malaria, we prefer to use a lot of transmission networks, most especially if you want to understand the genomic epidemiology and the transmission of the parasites. And also it can help us to get sinks and sources of the parasites so that resources can be allocated to combat malaria in the right places. And finally, the same panel is used to understand importation of new parasites in regions where malaria elimination has happened, or regions which are very close to malaria elimination in that case. So this tool called MadHatter and its wet lab panel was designed in collaboration with the Paragon Genomics company in US, and we are calling it CleanPlex amplicon sequencing. So to go in details about the panel, so we have, the panel is having diverse loci, which are about 165 loci for diversity. And then it has, like I mentioned, the non-falciparum plasmodium markers, which are 4. And then the drug resistance markers, both SNPs and the copy number variations, which are 15 genes being targeted. And then we are targeting 3 regions for diagnostic resistance, that is HRP2 and 3 deletions. And then the other is the CSP gene. And we put this in terms of primer pools, which we call amplicons. So there is a D1, which is the diversity panel. And then we have R1 and R2, or R1.2, which are mainly the resistance panel plus the other diversity markers. So primer pools amplify targets in 5 categories, like I've mentioned. And the ones for R1.1, it's basically the original pool, which was designed to increase sensitivity. But then we reduced it to a smaller version, which is what we call the R1.2. So in laboratory, I'll mention this because I know this is going to be about the tool itself. But in laboratory, we recommend to use a 2-multiplex PCR to increase sensitivity for low parasite samples. And basically, this is like take up DNA and then do a fast multiplex PCR where we combine diversity pool 1 and resistance pool 1. And then we combine it with resistance pool to increase sensitivity. And then we do indexing of the PCR, indexing PCR, and then do some cleanup like bead cleaning and all that before we do sequencing on any Illumina machine or Oxford Nanopower, depending on which platform you are using. So looking at the 14 chromosomes, the parts which are being targeted, or the chromosomal locations of all targets in Plasmodium falciparum genome, they are the ones you are seeing there. The purple is for diversity, pink is for drug resistance, green is for diagnostic resistance, and the red is for other species, and the orange is for the CSP, which is for vaccine. So you can see that we target different parts of the chromosomes, as you can see there. And the diversity, the diagnostic resistance category basically includes the targets around the HRP2 and 3 deletions, as well as targets in chromosome 11 that are often duplicated when HRP3 is deleted. So looking at the primer designs, I'll not go so much into the detail of the wet lab, but briefly, the primers define regions of interest from available whole genome sequencing data. mainly microhaplotypes of high diversity within African samples. And it also defines SNPs of interest, mainly drug-resistant SNPs across. And it also defines genomic regions to capture gene deletions for diagnostic resistance, and it adds species identification. So the primers are basically to amplify regions of about 225 to 275 base pairs in the chromosome. So going to the pipeline, so basically, briefly, that is all about the design. However, it's a little bit a lot. I just didn't want to go into the details of it. So I wanted to focus on the bioinformatics pipeline itself. So the pipeline basically starts with the output of the MySeq or the output of the sequencer, which is usually FASTQ files, which are then go into filtration or filtering and the multiplexing using cut adapter. It's a common tool that many people are aware of that does streaming and all that. This is mainly to filter primer dimers and demultiplex by locus, and also trim any other unnecessary parameters. So after that, then we use per locus filtering to look at the quality reports. So these quality reports are mainly showing coverage. They are mainly coverage reports, and they provide figures and tables for quick assessment of data quality, most especially to make decisions in the wet lab, as we shall see later. So after that, the pipeline sends the data for array calling. Here we denoise the data using DADA2. DADA2 is a common pipeline which is used for amplicon sequencing, most especially for those who understand metagenomics very well. So this is the pipeline we got, and we tried to pick out interest of how to play with it to make sure it can also do array calls for our data. So it's more optimized for multiple amplicons. So here we use inference of real sequences, and we use a couple of error models to merge and concatenate the reads. So after this, the amplicon sequencing data is put into a variant table, which we take into post-processing filtering. So this is mainly, we are doing a rearrangement of data, masking low complexity regions. Sometimes we call them homopolymers. For example, in malaria, we have a lot of homopolymers like 80-80-80 regions. It's 80-80 rich kind of genome. So we have to look out for that and also try to mask any other regions which did not sequence well and all that. So we call it post-processing filtering. After which, now we create the pipeline creates what we call an array table, which is containing the different microhaplotype data, which can be used now for downstream analysis. So the other thing to notice that this pipeline is put together in the next flow language because we are using multiple tools. Others have not mentioned them, like FastQC and others, but we use multiple tools for example, we have tandem repeat finders for those tandem repeats, and there are so many other tools which look at the complex regions. So we put everything together in the next flow language, and then afterwards we containerize the pipeline in 3 versions. So someone can use a version which is containerized in Aptainer, and another person can use a version which is containerized in Docker. And also we have a version which is containerized in Conda. And all these versions can be found down there on our GitHub page of the Epicenter. And the pipeline, like I said, it's called MAD4 which I explained earlier on. So like I mentioned, the coverage reports are mainly a couple of graphs and a couple of figures focusing on the read coverage, basically to help the wet lab make decisions. And these decisions are mainly based on, for example, this table you can see here is a report which shows the coverage by pool. For example, if you're expecting 165 from pool A or the diagnostic pool, and then you get 151, then it shows you the number of amplicons which have been sequenced on that specific pool in that sample, and so on and so forth. And then there is also another report which comes out, which is looking at the frequency and the versus the read count. And then that one also is used most especially to understand if the coverage was really good. And then we have this graph here, which is the third graph, which is also per pool, whereby it can show you how many pools, for example, what's the number of amplicons that have 100 reads and above, because we know that we define a SNP based on the coverage of the coverage of each amplicon. So this is helpful because it's used to assess the sequencing quality, most especially the evenness in coverage. We want to see that all samples have almost the same coverage and above, and then the modules, whereby the groups, like I said, there is module 1, which is diversity and resistance 1, and then there is module 2, which is resistance 2. So we want to see if both of them have a right evenness of coverage, and then the amplicons themselves, to look at each targeted amplicon to see that it has the right coverage, which can move forward. So it also helps us to assess the sequencing quality of the positives and negative controls. And if you don't expect any amplicon in the negative control, and then you see a couple of amplicons popping up, then you realize there was sort of contamination. And if positive control, you expect specific amplicons and you don't see them, or you see amplicons you don't expect from certain positive controls, then it will show you some sort of contamination. So the idea here is that the wet lab can use these reports to make decisions, most especially on sample sequencing, reprepping, and repooling. So if a sample is really badly injured, most especially in terms of contamination and all that, we can decide to just do a reprep and start afresh. And if a sample maybe just has low coverage, but most of the other parameters are okay, the run can be repulled, the samples can be repulled and resequenced in that way, but you don't have to start from scratch by reprepping it, since it's all about coverage. And it also shows us the possible sources of errors in the data set and in the analysis. So basically, that's mainly the pipeline. So what have we done so far with this pipeline to test its experimental performance? So the pipeline, the Mad Hatter pipeline, targets high diversity microhaplotypes, and we have compared it to 2 other pipelines which were in existence for malaria. So for example, there is the AmpliSec pipeline, and then the spot malaria pipeline. And if you can look at the medium heterozygosity, the number of our medium heterozygosity is 0.64, compared to 0.65 of AmpliSec and 0.5 of spot malaria. And if you look at the number of targets with the heterozygosity above 0.5, you realize that our pipeline, Mad Hatter, has 133 targets compared to 84 in AmpliSec and 55 in Spotlight, which gives it a high edge of capturing more targets with high heterozygosity in the Plasmodium falciparum genome. And when we looked at the resolution to try to look at PCA, we tried to principal components. We tried to compare how well can our panel or our pipeline distinguish between different data sets from different regions of the world compared to African as well. And you realize that what you are seeing in blue, or dark blue and light blue, and then some are brown. So dark blue is Central and West African data, and light blue is South and East African data. And then pink is South and Southeastern East Asia, greenish or dark green is Papua New Guinea, and then orange is South America. So you can see that if you look at, for example, spot malaria and amplisec, they couldn't clearly distinguish with high resolution between data from Central and West Africa compared to that of South and East Africa. But if you look at the Mad Hatter panel, the Mad Hatter panel tries on the one on the left tries to clearly distinguish between that compared to the other pipelines. So then we tried to see if it can also distinguish supranational data based on supranational level. So we have data from national level like Namibia, North Mozambique, South Mozambique, and Zambia. And the PC shows here, the principal component shows here that with the amplisec and the spot malaria, you cannot clearly differentiate or have a difference between North Mozambique and South Mozambique, but our panel could clearly distinguish between these two data sets, showing that it has a higher resolution compared to the other two panels which were in existence for malaria. Thirdly, we tried to look at how best power is used to infer relatedness or infection relatedness. And here we focused on data from microhaplotypes, where we delved into identifying relatedness of strains between infections, which is enhanced by highly multiplexed microhaplotypes. And the orange is spot malaria, green is amplisec, and the purple is Mad Hatter. And when you look at in incidences of multiplexed of infection where it equals to 1, that is a monoclonal infection, you realize that at different relatedness, Mad Hatter still had a higher edge of identifying to infer infection relatedness. And in incidences where the MOI is equal to 5, which is supposed to resemble a polyclonal infection, you still realize that Mad Hatter is outcompeting all the other panels at the different relatedness levels of this. And if you look at the SNP data, you also realize that still it's the same thing. The first one is microhaplotype, the second one is SNP. So if you look at SNP data, you still see that even with the SNP simulated from whole genome sequencing with the different allele frequencies, different population allele frequencies, still the data was showing that compared to reconstructed microhaplotypes, the data are showing that Mad Hatter still had a higher age compared to the other three panels, the other two panels, amplisec and spot malaria, which have been in existence before Mad Hatter came in. And also we tried to check reproducibility. And here we tried it on different pools. For example, you see that down here the first graph here you are seeing is we tried out a number of samples with the different parasite densities, which we call parastemia, which is parasites per microliter in a sample. And you realize that even with less than 10 parasites per microliter, the different pools are being fully sequenced compared to the others. And also as the parasite per microliter or parasitemia increases across the different samples, you realize that all pools for diversity resistance and diagnostic markers are all being amplified well with good reads. So if you look at the second graph there, it shows the proportion of targets with more than 10 reads in the DBS, which is dry blood spot controls. So the first column has 1 parasite per microliter, and the second one has 10 parasites per microliter. And it shows the third one is mid-gut parasites. When I say mid-gut samples, I mean we know that Plasmodium falciparum has two hosts, the human and the vector, which is the mosquito. And in the mosquito, recombination happens in the midgut of the vector, which is the mosquito. So we got samples from the midgut, samples of the parasites from the midgut of the vector, and we are showing here that all of those parameters, including the different parasite densities, very low, like 1 parasite per microliter is quite very low, but we still pull a good target of the amplicons coming up. And we are showing here that higher parastemia still strongly correlates with improved sequencing, depth and completeness. And we are also seeing in the second graph that at low parasite densities, as you can see 1 or 10, the panel still pulls out a good number of higher detection for this data. So continuing with the sensitivity, we also checked sensitivity from the genetic data of varied samples or variety of samples. And we are looking here at the allele frequency or allele recovery versus the expected within a sample allele frequency. And we are seeing that both for alleles and the allele data, which is from the diversity panel, and the resistance panel, we are seeing that the pickup is quite good. Recovery within the sample allele frequency compared to the diversity, it is 161 loci across the 183 samples. And you see that despite the varying parasite densities, as you can see, less than 10 in orange, green is 10 to 100, pink is 100 to 1,000, and the blue is above 1,000. You can see that the data is having good recovery across the varying parasite density. And on figure D, which is showing the recovery in biallelic SNPs in the drug resistance markers across the 20 codons, it is showing that the data is still shows that the recovery was quite remarkable. But most especially what we look at here is that the recovery is higher at higher parasite density. As you can see, the blue is almost a perfect curve. And then the recovery goes on reducing, the power goes on reducing as parasite density reduces up to less than 10. So both panels show that higher expected within sample allele frequency leads to better recovery. And this is modulated by parastemia. And high parastemia samples, like I've mentioned, they have consistently high recovery rate, which is making them more reliable for allele frequency and resistance mark analysis. So if we look at this further analysis, we tried to see how much is observed within sample allele frequency versus the expected. And we are seeing that our panel or our pipeline has a high sensitivity. Though if you look at the first graph in the corner, it's for less than 10 parasites per microliter. The second one is 10 to 100. The one down is 100 to 1,000, and the one in blue is above 1,000. And we are seeing that the R squared goes on increasing as you increase the parasite density. So if you are using data with above 1,000 parasites per microliter, you are likely to have the highest sensitivity from this panel. So MADHATA demonstrates here a robust and accurate estimation of frequencies or allele frequencies in mixed infections, particularly in high parasite densities, though the data from low parasite densities is also still reliable. And this shows its ability to detect minor clones accurately, making it an effective tool for molecular surveillance and the complexity of infection analysis. And here we tried to show, to demonstrate how MADHATA yields highly reproducible within sample allele frequency values across multiple global labs. So the first graph there shows that we mixed data from different labs, about 7 labs. There is the purple, which is IS Global. In Spain, there is data, I think green is IDRC, Infectious Disease Research Collaboration from Uganda. Yellow is NICD, which is National Institute of Communicable Diseases from South Africa. Then blue is a mixture of samples from IDRS and IS Global. And then the others are mixture of the different samples with those from University of California, San Francisco. And the end of the day, we tried to show, the panel was showing that multiple global labs can use the same panel and the same pipeline and still achieve standardized genomic surveillance results. And the second panel here shows that Mad Hatter accurately reflects population level genetic diversity. As you can see that we are having a good kind of correlation between the expected heterozygosity and the observed heterozygosity. And this is very good when you are doing population genetics differentiation at that level. So then here we looked at the screening of deletions and duplications. So like I mentioned, this panel can screen deletions, HRP23 deletions, and then duplications, which are mainly for copy number variations. And we are seeing here that Mad Hatter accurately estimates gene deletions and duplications based on read depth normalized to control samples. So we can see that the pools, for example, in graphs in figure A, the different pools like the diversity pool, resistance pool 1 and resistance pool 2, all of them are having a good read accuracy or a good read coverage for the different amplicon sizes being sequenced or being targeted. And we are seeing that in part B, we have a good fold change of the different HRP2, 3 deletions and MDR1 gene capture with the different parastemia or parasite density levels. And this method shows that it's robust across replicates and performs well in mixed infections at different or variable parasite levels, which makes it suitable for high throughput molecular surveillance of diagnostic gene deletions and resistance gene amplifications in field samples. So a couple of countries are currently using this pipeline to do their analysis, most especially malaria molecular surveillance. I will show you a couple of countries. So far, we have a few papers which have come up, published using this exact pipeline, including that paper from March 2025, which is sensitive and modular sequencing of Plasmodium falciparum diverse and resistance for research and public health. This is the paper for the pipeline, which is in scientific reports. And then we also have researchers from Southern Laos who have also used this paper to understand genomic epidemiology of spatially clustered local transmission of Plasmodium in forest-going populations in Southern Laos. And the other one is performance, where we tried to compare. A colleague of ours, Thomas Katairo, led us to try to compare the performance between the resistance, the panel known for resistance markers, or to understand resistance markers called the MIPS, or using molecular inversion probes, and the hours of Mad Hatter. So we are trying to compare a lot of performance metrics in this paper using Plasmodium falciparum mutations which are associated with antimalarial drug resistance. And we found that the panel was really performing remarkable. If you're interested, you can look out for the paper. So about implementation, this panel, like I mentioned, it is being used in a couple of labs across the globe. So these are some of the labs. It is being used at University of California, San Francisco, where mainly it was developed, but still being used. and also at the Chan Zuckerberg Biohub. It's also being used at a lab in Yale University where they are trying to understand malaria drug resistance. And also it's used at IS Global in Spain. There is MRC unit in Gambia, and there is also Institute Pasteur in Dakar, Senegal. And down here, it's being used at the National Institute of Communicable Diseases in South Africa. They are using it in Mozambique, in Uganda under IDRC, and Ethiopia under AHRI, and also in Southeast Asia. So you can see that the panel is being implemented across different labs. And we even have a collaboration, which we call the Tea Party. So basically, most of the labs which are using this panel, and the scientists using this panel, they always come together in a meeting called the Tea Party, and we discuss how the panel is performing. And it's basically like a community we created where we're trying to understand challenges in the different data sets and how to navigate, how to solve them using the panel, the Mad Hatter panel. And we're also doing a lot of capacity building where different experts go in different countries to try to see how the panel is being implemented. As you can see the pictures there, the photos there for the different labs, and they're trying to implement the panel and going through training. Like I mentioned, we have a well-elaborated laboratory protocol, which is, as you can see there, it's called the Mad Hatter protocol, which you can easily find online. And we also have the bioinformatics pipeline, which you can get from GitHub, as I shown earlier, where you can understand its installation and usage. And we are always available to help or assist in any way. So in summary, like I said, the panel is called the Mad Hatter panel, which is an essay which is modular and multi-purpose, targeting about 165 diverse loci with high geographical resolution and the power to identify related infections, targeting about 13 drug resistance marker genes, including K13 and the CRT and others. It targets the HRP2 and 3 deletions, which is the diagnostic resistance markers in most of the RDTs used across Africa and countries, and also the CSP gene, which is mainly for a vaccine, a target for vaccine. So the bioinformatic tool, basically, it's known for its portability and wet lab decision making. As I mentioned, that it has a couple of outputs, which include a couple of reports where a wet lab team or the laboratory team can make decisions on whether to repull or reprep samples or resequence if the worst comes to the worst. And we have quite a lot of training materials we can use for people who are implementing this pipeline. So I would like to acknowledge a couple of people. As you can see there, this pipeline was created, the contribution is tremendous by so many people, but particularly it was led by Andres Aranda Diaz, Brian Greenhouse. Andres was at UCSF, now at IS Global, and Brian Greenhouse is a professor at the Epicenter at UCSF. Nick Hathaway, Eric Brian Palmer, Catherine Murray, who are the senior bioinformatics scientists, Ina and Emmanuel and Debayan and Edward, and a couple of other people whom we worked together tirelessly to create this pipeline. And a couple of institutions. Personally, I sit at the African Center of Excellence in Bioinformatics and Data Intensive Science. And I'm also part of the DSI Africa. That's why I'm presenting here. Under the MAC Data Project in Makerere University. But a couple of institutions like Chan Zuckerberg Biohub, Africa CDC, funders like Bill and Melinda Gates Foundation, NIAID and NIH, and the IDRC, also an institution where I sit as a genomics research fellow. So all of us have really contributed to the development of this pipeline and make sure that it's being delivered timely. Thank you so much. I welcome any questions. Back to you, Rolanda.
Sure. Thanks so much, Said, for for this very interesting presentation. Let me see if there's any questions from the audience. You are welcome to unmute and ask your question, or you can type it also in the chat. Okay, while folks are thinking, I'll ask a question.
I don't know if it was a lot. I know it's a lot of, there is a lot of malaria and
So I have a question while the others are thinking about their questions. So I
yes.
noticed on the map when you were showing where the tool had been adopted, Latin America, that also has some malaria outbreaks, they are not, are they involved at all in also trying to adopt this tool? Or what do you think might be the challenges? Why? Is it because the prevalence is really low there? But I don't think so, because apparently in the Amazon basis there around is quite large number of infections. So just any thoughts on that?
Yes, so so Latin America has quite a lot of malaria, like you have mentioned, and before Latin America is actually using a lot of spot malaria, one of the panels I was comparing with. So that's what they have been using previously, and also most of them have been sending their data to Sanga. Sanga Institute in UK, because Sanga has a well-established malaria surveillance program under the malaria gene. So most of them have been outsourcing their services. However, during a couple of conferences, we have presented this work and a couple of other outputs of this pipeline. Like the last year, we were in GEM, genomic epidemiology of malaria in UK at Sanga Institute. And also we had the MIM, which is the malaria multilateral initiative on malaria, which was in Rwanda last year. A couple of guys from South America came in and they appreciated this tool. And right now they are in the process of starting to use it. Yes.
Okay, no, that's good. Okay.
Yeah.
Thanks for that.
You're welcome.
Anybody else with a question? There should be. So also then, while I still have the floor, are there any ideas of how to adopt this tool maybe for other vector-borne diseases or vector-borne parasites, like the closely, or not closely related, but the similar transmission dynamics, like I think chikungunya disease is also very prevalent probably in South America. And then, of course, the trypanosomiasis. I don't know if you have.
Yeah, so what I can say is that when we created this panel, it was mainly designed with malaria in mind, and it's based on reference genomes of P. falciparum and the other 4 non-falciparum species of malaria. So I think it's malaria specific. I don't think it can be applied to other vector-borne diseases yet.
But could you follow a similar protocol to design something like this for one of the other parasites, in your opinion?
Exactly. Like when we are designing this, actually we based the idea from metagenomics of bacteria, where
Okay.
you are targeting, for example, 16S amplicons in bacteria in metagenomics. So the idea came from bacteria, and then we evolved it and we implemented it in malaria under this panel. So I'm sure someone can use the same exact idea to create a panel for any other pathogen.
Okay, well, that's great.
Yes.
I see
The principle behind is targeted amplicon sequencing. So for as long as you are going to do that, you can always borrow the idea.
Yeah, that's good.
Yes.
Very good example. So Ulianka has I don't know if Ulianka, if you want to unmute and ask your question.
I'm seeing some comments. Olainka Adebajo is saying, I just wanted to say that this was a great presentation. I have heard of falciparum before, and it's quite amusing to hear you talk about it and share the designed bioinformatics pipeline. Are there open source outputs for secondary analysis? That's a great question. Thank you so much. So yes, so one of the current, after designing this pipeline, actually now the target or all the energy is trying to have what we call standard outputs across different laboratories doing malaria across the globe. And we have a current project funded by NIH going on, and Bill and Melinda Gates. So we are trying to create a sort of a database, an online database, where data from different countries can be put together. And if someone wants to look at a general picture of, let's say, a region, they can just find the data from the multiple countries within that region from the same source. So that is on underway, and it's being worked on right now. So probably we shall let people know when it's done and it's available for use. But thank you so much. Yes, I'm seeing, oh, it was the same comment. Thank you. Yes, Rolanda?
And I was saying, if there are no more questions, I don't know if anybody, let me just give a second. If there's any other questions from the audience, you're welcome to unmute yourself and ask a question. All right, I can't see anything at this moment. All right, then. Thank you so much, Shahid. I think really it was very interesting. I think the folks will still mull over your presentation, and then hopefully they can reach out to you to further tap into your expertise for this research area. So if you're happy, then we will share your slides, if that's okay with you.
That's very fine. I'll share them with you, and then you share with the rest of the team.
For sure. Then thanks so much, everyone, for joining today, and thanks, Shahid, for making the time to present this interesting work to us. So we'll see you guys on the next webinar.
Thank you so much, and have a good day.
Thanks so much. Thanks, everyone. Have a good day further. Bye.